So you used a supervised learning model for predicting how people would probably taste one of your tens of thousands of simulated recipes. Yeah. So how's that coming along?
It's coming out amazing. It's so unbelievably smooth, really light, and out of all the lagers I've ever made, it's up there. It might be the best, like the best lager I've ever made. I was like, this is so bizarre, but in a really cool way. I feel like there's like an alien telling me what to do, and it makes sense.
Beau, welcome to the Super Data Science Podcast. It's so cool to have you here. For people who are watching the YouTube version, they'll see me for the first time ever on the podcast. I'm having a beer on air, and there's good reason to be doing that because you're a brewer. And tell the audience how we met, Beau. How did this all come about?
So I was walking my dog. I remember that specifically. I'm trying to remember. I think it was in like 2022. Maybe it was 2023. And I already had this idea to really utilize AI and data analytics to create beer. So I already started on the project. I was listening to episode, I forget the episode. I know you'll be able to help me out with this one.
It was the episode about beer. So it was episode with Zach Weinberg on like using ChatGPT for it was like the layman's guide. So yeah, so then the context here is it was early 2023. So that episode came out on January 20th, 2023. It's episode number 646. And in that episode, my friend Zach Weinberg, he's one of my oldest and dearest friends, him and I had been talking, and his mind was so blown about ChatGPT, which at that time was brand new, and he was using it for tons of purposes within his brewery to make it easier to create copy, to create marketing materials, to answer questions in an automated way that people might have, particularly on his website. And so, yeah, he came on the show to do this episode. It was a Friday episode on ways that non-programmers, lay people, I guess, could be taking advantage of AI tools like ChatGPT to automate things. And yeah, so I guess that's the episode you're referring to. That was about a year ago, episode 646.
Yeah, so I was listening to it. I was like, I'm literally doing that, like, because I know he mentioned, like, someone should get all these feature columns together.
Right.
You know what I mean? Yeah, I was like, I'm literally doing that right now.
Yeah. So, he's like, here's an idea. Yeah, I think his idea in the episode, if I remember correctly, he said something like, because at that time GPT 3.5 was trained on everything on the internet, there's tons of brewers who just publish their recipes. Yep. So he was like, you could be using the knowledge embedded in this large language model in order to get recommendations on how to brew a recipe. I don't think it was as specific as what we're going to get to later in this episode with like feature columns.
Gotcha.
But definitely he talked about the idea of ways you could be using LLMs to generate beer recipe suggestions, if I'm remembering correctly. Yeah.
No, I think you're right. So, I mean, I was doing something similar, I guess. Not literally that. So I was like, man, I got to reach out to Jon. So remember, I hit you up on Twitter, crossing my fingers, and you hit me up, and you're like, dude, let's chat. I was like, let's. So, yeah, just kind of went from there. And then one thing led to another, and all of a sudden we're doing a collaboration beer together. And I'll tell you what, it's I'm really excited how it's come out.
Yeah, we're going to do the big reveal later. So to kind of give an overview, we started working on this collaboration, or we started talking about potentially collaborating a year ago. That picked up in the fall, the Northern Hemisphere fall of 2023, as you started to get a new brewery together called Species X. I don't know if you want to tell us a little bit about that, because that's also people, even our audio only listeners, will be able to hear kind of the rich echo of this massive space that you're brewing in. And people watching the YouTube version can see this epic background behind you. So yeah, do you want to tell us about Species X Brewing?
Yeah, so Species X started out in, I think it was 2021, at least the idea came around. And the whole idea was, this is before the pandemic started. I had this idea. I listened to a lot of scientific podcasts. So one was about microbiology. They were talking about finding the next pathogen. How do we prepare for the next pathogen? And
that was before COVID.
That was before COVID. And they called it Pathogen X. So I was like, I would love to do that. Like, I would love to find the next great organism or species in beer. And that's how I came up with Species X. Now, granted, AI is a little tongue in cheek. People say that could become the next species. So I'm kind of taking that, not, super seriously, but as, part of the brewery. And then the other part of the brewery is all novel biological organism focused. So genetically modified yeast, force mutated yeast, wild captured yeast, spore germinated yeast, just all things biological and novel in nature, at least to humans. So we have those two separate realms. We call the silicon species, or the silicon species is the artificial intelligence ML models that I've created and coded, and then the other is the carbon species. So that's going to be your GMO yeast, novel biological organisms. So,
gotcha, yeah.
Yeah.
So Species X, I've even seen that on your website when people go to buy beers, which will be easier and easier. Right now, it sounds like there's only a couple of beers available at Species X, at the Species X website. And those are maybe at the time of recording only available for shipping in Ohio.
We do not. So those 4 beers online are collaboration beers. So they're only available at Aslan.
Where you used to work. I see.
Yes. Yeah. Yep. We will have 4 flagships. So 4 flagships will be available through distribution in Ohio. And then, so we have an alternating proprietorship brewery in Virginia that will be brewing that for distribution. And then we'll be doing pilot batches here. So little 3.5 barrel batches of beer, which equate to 7 full kegs of beer.
All right. So what you're saying is that at the time of recording, there aren't real Species X beers brewed at Species X that people can yet buy online, but it's going to be happening more and more as the brewery opens more and more. It's something that's just happening. So you've been kicking off the Species X brewery, and the main thesis that you have as president and I guess head brewer of Species X?
Yeah, head brewer. Trying to think of all the hats I'm going to be wearing in here. I'm the only production person president. So I'm, taking care of costs and a lot of managing all of our employees, looking at costs and P&Ls, that sort of thing. Really exciting stuff.
So the thesis there, as president and manager and P&L head and production person, all these different hats that you're wearing at Species X, there's 2 main theses. One of them is that novel biological organisms like GMO yeast can produce interesting beers in a way that no existing yeast could or no other brewery could. And so you call those your carbon species?
Yeah, correct. Yep.
And then you also have your silicon species, which is mostly what we're going to focus on in this episode, or eventually we'll get to focusing on quite a bit.
Correct.
And
yep.
so You've written a ton of code. I have I've seen the code. I haven't worked on it myself. I haven't written any code, but I've reviewed some of your code and kind of had some ideas of ways that you can make changes potentially to data that you're including, or ways that you could be processing or modeling the data. And so we're going to talk about all of that silicon stuff later in the episode. We're going to talk about how you have been using machine learning in those silicon species to create what could be some of the greatest beers ever. And I'm going to get to find out on air how our first beer is coming along. So we're going to get to that later in the episode. But first, let's talk about beer in general. So you are a literally certified beer expert. So you're something called a cicerone, so people who go to fancy restaurants, they may be aware of the job title sommelier, which is somebody who is a specialist in wine tasting. Um, and the equivalent in the beer world is a cicerone. And I've only read about this in The Economist. So I don't know a huge amount about cicerones, but I know that you are a level 2 cicerone. So maybe you can tell us a bit about that. And another big credential, other than your many years, you have about a decade of experience working in brewing, but you also have studied microbiology at the Siebel Institute in Montreal. So tell us a bit about how somebody becomes as expert at beer as you, and what these kinds of certifications like the Cicerone mean, and why it would be useful to study microbiology as a brewer.
Yeah, so obviously I'm obsessed with the art of brewing. In addition to the art of brewing, I'm absolutely obsessed with science. So a cicerone, this was back before I stepped into full-time production mode. I worked at sales for several companies. One of these companies allowed us to study to be a cicerone, through the cicerone program. And we had a pretty intense 2 weeks studying that. Level 1 is certified beer server. So you can actually take that online fairly quickly. It's still very hard. So you definitely need to study for it. Level 2 is exponentially harder than that. So I believe, from what I remember, it's about a 5 to 6 hour test. There's a tasting.
A 5 to 6 hour test. Do you get to do you get
I
a
hope I'm right
break?
about that, but I'm trying to remember because it's so long ago.
You were drinking at the time.
There was a sensory portion, which didn't take too much time. Yeah, sensory portion.
So level 2 is obviously in person. It kind of was implied by you mentioning that level 1 could be done online. And then, so if there's a sensory portion, then that must be done in person.
Yep.
That's cool.
And then there's like a 3-hour written portion. So maybe it's more like 4 to 5 hours.
It doesn't matter. We're getting the rough idea.
But yeah, anyways. Yeah, so that was really hard. I found that there's some off flavors that I just do not have the right threshold for it. So I had to take the test twice because of the sensory portion.
Oh, really? So you aced the written part, but the sensory part was hard. Data science and machine learning jobs increasingly demand cloud skills, with over 30% of job postings listing cloud skills as a requirement today, and that percentage set to continue growing. Thankfully, Kirill and Atle, who have taught machine learning to millions of students, have now launched CloudWolf to efficiently provide you with the essential cloud computing skills. With CloudWolf, commit just 30 minutes a day for 30 days and you can obtain your official AWS certification badge. Secure your career's future. Join now at cloudwolf.com/SDS for a whopping 30% membership discount. Again, that's cloudwolf.com/SDS to start your cloud journey today. And that's amazing, because you obviously, assuming that you did this certification relatively recently, you already would have had a decade of experience professionally tasting beers. So that really says something about this taste.
No, I I was in the industry for probably 3 years by then. So this is, like I said, this is pretty far back ago. But I just, some people just don't have a really high threshold for things, and I don't like there was like 1 or 2 things that I just had struggled picking up. So, anyways, so I got that, which I was really excited about. Beyond level 2, there's also advanced Cicerone, and then there's master Cicerone. Advanced is also exponentially harder than level 2.
Oh.
So, and then after advanced Cicerone, that's exponentially harder than that. So I have nothing but the most respect for not only the people taking level 1 and 2, but seeing how I failed the first time on getting Cicerone level 2, how hard and dedicated you have to be to achieve advanced Cicerone and to get the master Cicerone, which is, I think there's only a handful of people in the world that have master Cicerone.
That must have been what I was reading about in The Economist. It's like dozens of people worldwide that have that. Yeah, that's wild. And so there must obviously be some kind of, maybe not obviously, there might be, I suspect, a strong like biological component to being able to do this. Like my sister, for example, she could just taste way more stuff than me. I've never been a great taster, and I think we actually looked into this growing up. There's like a density of taste receptors on your tongue, and people have different densities, and so you can get like a little. They have these little. They're like the size of like a Cheerio, a flat piece of paper that looks like a little Cheerio. You can put that on your tongue, and you can count how many taste receptors are in that little Cheerio-looking circle thing.
Really?
And that gives you a sense of how many taste receptors you have on your tongue, and some people have way more than others.
That's fascinating. I did not know about that. Start handing those out.
Yeah, you could. You could look that up, and it's something you can buy. It could be kind of an interesting, actually, if people are coming by and doing a tasting at the brewery, you could have these there, and because it just takes like a minute to count, and then it gives you a sense. There's like super tasters, which my sister is because she has a crazy number of taste receptors, and I'm like, I can't remember what the word is for like a sub taster. I'm like deficient.
Yeah, it's funny. My wife is the same way.
She's a super taster.
Yeah, super taster.
Yeah,
She's
you'll have to
really
do
good.
the test to see whether it's experience or biology in her case.
Yeah.
Cool. Yeah. So that's the Cicerone. And then, yeah, in addition, you studied microbiology. So I guess we're going to get into how beers are made in a moment, but I guess people might not appreciate the extent to which brewing beer involves working with living biological organisms. And so microbiology training can also be a huge asset. Do you know many other brewers that actually formally study microbiology like you did?
I'm trying to think here. Obviously, the bigger brewers around are gonna send off the right people to go get trained. Obviously, all lab techs, you definitely want some sort of lab training, which is prominently why Aslan sent me for 2 weeks in Montreal to go study beer microbiology.
Right.
I was the head of the lab, so I did all the QA/QC, microbiology checks, all that stuff.
Yeah, I'll explain a few terms here. So Aslan, that you referred to a couple times now in the episode, that's a brewery in Alexandria, Virginia, that you worked at for 5 years. And when you say QA/QC, this might actually be obvious to some software people out there because QA is also software engineer. It's like a software engineering kind of specialization. It's a quality assurance. So the QA/QC is quality assurance and quality control supervisor. You were, and yeah, I see. So as part of that role, you're spending a lot of time. Like, are you literally like working with like test tubes and that kind of thing?
Yeah, I was making plates. We had a nice set of selection for our plates. So
plates for growing like a bacterial culture kind of thing.
Exactly. Yeah. So what I love to do was my favorite plate was WLN because it would change colors depending on the yeast strain, the species of yeast, the bacteria, all that stuff. So it made identifying colonies a lot easier. So that was like my go-to general media if I'm trying to troubleshoot something or if I captured something I was interested in it. That was my go-to. And
So
then we have what's
when
up?
would you do that? Like when in the process? Is this like during the brewing process? Would you just if something looks unusual, something looks like it's a rye or tastes like it's a rye or smells like it's a rye, or you just are brewers kind of always doing these kinds of tests, plating yeast and bacteria, I guess. Is this like when does that happen in a beer brewing process?
Well, I think ideally at every single step, so going into the fermenter, you want to make sure that wort is sterile and you have a certain CFU, cell forming unit on your plate.
And what's the wort?
Oh, sorry. Wort is like the
Is it like
sugary, it's like sugar water and hop. It's sugar, water, or it's sugar from the grain. Any hops if you use, well, yeah, all beer pretty much has hops in it. So hops, unfermented beer, I want to say. So
got it, got it.
it's like sugar water essentially.
It's spelled WORT, if I remember correctly.
Yep, that's correct.
And this is what you start with. So you as a human can mix together kind of your first set of ingredients in water. And yeah, the key ingredients in beer, I guess, are barley, malts, hops, and water. Right? That's typically what you would put in, those kind of four things.
Yep, and then people can throw in some adjuncts. So wheat, oats are used a lot in like New England IPAs or the hazy IPAs to increase mouthfeel. A lot of people use rye to add like a little bit of a spicy character, not necessarily in New England IPAs, but see it a lot in stouts and stuff. You also see a nice roasted barley for stouts. Guinness is well known for using roasted barley. You'll see some other kilned malts to darken the color, but also not add, and they'll caramelize it. So it won't add like a roasty bitterness. It'll actually add like caramel or caramel for some people.
Okay. So
yeah.
so all these different things are added together into water. It's kind of like a sugary sweet water, and that's called the wort. That's, I guess, the starting point for brewing, and then you add yeast to it to start fermenting the sugar water into alcohol and carbon dioxide.
Yeah, we start on the hot side. So as you mentioned, you make that sugar water, boil it for an hour, or some people will do 3 hours. If you're doing like some super stout or barley wine or longer, and then you throw it through a heat exchanger. Immediately cool it down to fermenting temperature. Infuse it with oxygen for the yeast, and then pitch the yeast into the wort that will be in the sanitary tank or the fermenter.
Cool. And yeah, every step of the way, we could be doing like QA/QC, looking at how the yeast is doing, I guess, and what kinds of bacteria are present. So that isn't something
Yeah.
I'm really aware of. So both of these things are very small organisms,
Yeah.
and yeast, I know, is the animal that is actively, because if I'm remembering correctly, yeast is like the simplest. It's one of the simplest animals. It's a single-celled animal. So it's not a plant. It's not a bacteria. Those are like different kingdoms
Yep.
of animals or species. And so this tiny single-celled animal, the yeast, is adept at converting sugar into alcohol and into carbon dioxide. That's how it survives. But why are there bacteria present? It sounds like that's a pretty normal thing. To me, that sounds like potentially an alarming thing. Like you don't want a bunch of bacteria growth in your food typically.
Yeah, you definitely don't. And our processes at Asen were very good, so we never really saw a ton.
I got you.
And if it was there, it was under threshold, which is barely any. But yeah, if it's there, most of the time you don't want it there. Some of it can't tolerate alcohol or hops.
Right.
So what'll happen is you'll see it pop up on a plate, and it's not going to be able to grow in beer. So that'll happen sometimes.
Gotcha.
Now
So like,
that doesn't mean that like everything's perfect and dandy. You want to get rid of it, but there's other things like lactic acid producing bacteria that will sour your beer, and that is something that is like giant red flag, such as pediococcus, which is super small cell compared to a yeast cell. So that stuff, as you can imagine, can just embed itself into everything in the brewing process. And that can cripple a brewery essentially.
wow, because it's just kind
Yeah,
of, it becomes like a pandemic, like a bacteria pandemic in the brewery.
yeah, and I mean, unless
Uh,
like, yeah, I mean, like most places, luckily have checks in place and they're able to detect it and narrow down where it's coming from. But yeah, that's kind of, there's a lot of stuff going on in beer.
yeah, and like living stuff. And so sometimes things can get out of control, I guess. So we've gotten this sense now that the quality control involves monitoring for bacterial growth. Are you also monitoring as part of a QC process, literally things like flavor? Like, do you taste things along the way to make sure that things are kind of coming along, or what other kinds of quality control steps are there as you go through the process?
Yeah, definitely. And as we're about to talk about this project, the most important thing is if it tastes good.
Right.
Because, even if all your checks are perfect and you're hitting target and everything, nothing weird's growing on plates, your dissolved oxygen levels are great at canning. If the beer tastes like, then, it doesn't matter how it looks on paper.
There's it's completely bacteria free. Enjoy your beer.
Yeah,
I don't
enjoy it.
know. Who cares about the flavor? There's no bacteria.
Yeah.
Um, okay, cool. All right. So I'm starting to understand this a bit better, starting to piece things together here, hopefully for our listeners as well. So when you're brewing beer, one of the key things that people hear about is ales versus lagers. I think the popular perception, if people will say, I like lagers or I like ales. And I think that's because in the mass market, lagers are typically like relatively light colored beers. The one that I had but have now finished, it was a lager. So it was that light color. People can go back to the beginning of the episode to see when I had a full glass. And then ales are often on like grocery store shelves and in beer store shelves, like a darker color, and maybe they have a bit of a richer flavor, a sweeter flavor. So I think people think in their heads that that's what the difference is. But my understanding, and I've never brewed a beer in my life, but lager versus ale, it's really just to do with one of them is top fermenting, the other one's bottom fermenting, and I can never remember which is which. But um, you could actually, and correct me if I'm wrong, but can you basically get any kind of flavor out of a lager or an ale, or these kinds of distinctions of lagers being light, ales being dark? That's kind of like that's an oversimplification, right?
I think it depends. So, your macro lager, super clean, super light. It's pretty much got like zero sugar left because the yeast have pretty much eaten everything because they added it. Most of these places, these macro lagers, which we make these style lagers too, based on one of these models I made, but I'll talk about that later. They're super clean, easy drinking, no phenolic off flavor, no weird esters or flavors, super clean. And the reason they're clean is because of Saccharomyces pastorianus. And pastorianus is very efficient at fermenting at cold temperatures. And the reason Pastorianus is very good at fermenting cold temperatures is because you have Saccharomyces eubayanus, so S. eubayanus, and then you have Saccharomyces cerevisiae, which is your ale strain. These two, at some point, they made a hybrid, so they mated, and it must have been a really harsh environment, and then it sporulated and they created this new offspring called Saccharomyces pastorianus. So pastorianus that everyone uses is actually a hybrid strain. So what it did is it got its cold fermenting tolerance from its eubianus parent, and then it got its maltotriose complex sugar ability to metabolize from its cerevisiae parent. You combine those two, and you have a very efficient machine at low temperatures. And because it's at lower temperatures, it's going to be a lot cleaner, not a ton of flavors and esters going on. I like to explain that, like, the reason ales have more esters and flavors is it's kind of like an equivalent of if you're moving furniture and it's like 80 degrees or 100 degrees in your house, you're going to be sweating a lot. And the sweat, like the sweat equivalent is like the esters and flavors.
Eager to learn about large language models and generative AI but don't know where to start? Check out my comprehensive 2-hour training, which is available in its entirety on YouTube. Yep, that means not only is it totally free, but it's ad-free as well. It's a pure educational resource. In the training, we introduce deep learning transformer architectures and how these enable the extraordinary capabilities of state-of-the-art LLMs. And it isn't just theory. My hands-on code demos, which feature the Hugging Face and PyTorch Lightning Python libraries, guide you through the entire lifecycle of LLM development, from training to real-world deployment. Check out my generative AI with large language models hands-on training today on YouTube. We've got a link for you in the show notes.
Gotcha. That's super interesting. Okay, so the lager yeast, which you pronounced it there. I'm not going to. I would just completely butcher it. But the lager yeast. Say it again.
Pastorianus.
Yeah. It is able to ferment at very cold temperatures. Which, using this analogy, that's going to help me now remember that it's a really clean flavor, because it's like using Fahrenheit, which half of our listeners are in the US, so that'll make sense to half of our listeners. In Fahrenheit temperatures, when it's like 40 degrees, 50 degrees, it's a pretty cool day. That's about 5 degrees or 10 degrees Celsius. And at those kinds of temperatures, if you were to do a bunch of moving furniture around from one apartment to another or whatever, you wouldn't get very sweaty. And so that's kind of what's happening with these Pastoralis, this lager yeast. It's getting at low temperatures, it's able to do a lot of work efficiently, converting the sugars in the wort into alcohol and into carbon dioxide, giving beer its alcohol and fizziness, respectively. And so you're typically getting with lagers a nice clean flavor. So you're completely ruining my hypothesis around lagers and ales not necessarily having different kinds of flavors. But then ales, on the other hand, it's like moving on a very hot day, moving that furniture on a hot day. So, if you're talking about like an 80 or 100 degree Fahrenheit day, that's like a 30 degrees Celsius day. Very hot outside, you're getting really sweaty. So similarly, the yeast, they're just shedding all kinds of interesting flavors into the beer and giving ales, which I tend to prefer, they're yummy flavors in my view.
Now think about this too. When you're in a hotter environment, you're able to move around a little quicker and get stuff done faster.
The joints are all lubricated.
Yeah. So lagers are a little slower in most cases than ales can be. And then you also have to think about, just like humans, some humans are just predisposed to sweat more, even if it's like, 1 degree outside. You know what I mean? So some of these other strains, these cold tolerant strains, will still impart esters and flavors reminiscent of an ale strain. So eubianus, which is the parent of Pastorianus, tends to have they call it phenolic off flavor, just because it naturally expresses it due to its nature. So that's where it gets kind of confusing. I'm not going to dive deep into this right now, but yeah. So in a general idea, that's like the best metaphor I can really figure out for the understanding of this.
Yeah, no, that makes sense. I've definitely learned a bunch there. I've been misinforming people about lagers and ales. But one of them is top fermenting and the other is bottom fermenting, right?
Yeah, I'm trying to think. The only reason one is considered top fermenting is because there's a really thick rosin in ales, because all the cells they're flying all over the place, and it tends to foam up at the top.
So crossin is like a foamy layer on the top of the wort.
Exactly, yeah, and that's mostly cells up there, which is true. But lager strains, they don't just like sit on the bottom. They're all over the place, but they don't make like a big thick rosin like ales do.
I see. So it's a visual thing. So
exactly,
people call ales top fermenting because you're seeing all this on the top, all these cells bouncing around. Gotcha. But it's an oversimplification to call ales top brewing and lagers bottom brewing. Cool. All right. So that is super helpful for me. So now let's start to get into this machine learning project. So at a high level, the idea that you had, so from your experience brewing at prior to Species X, you have a data set of several hundred rows where every row represents a different beer, where that beer was went out to the public and the public tasted it. So for each of those hundreds of rows, you have a couple hundred, well, I don't know, maybe not a couple hundred, several dozen columns of information. And those things relate to things like what was the alcohol level, which is kind of easy to understand. So I'm looking at one of these spreadsheets right now, and so there's some beers have as little as 2%, some have 7%, some have 10%. But there's lots of other features in the data set related to ingredients that went in, that things that relate to the process that was followed, like temperatures that were used, ratios of different things to each other in the brewing process. So it provides this really rich feature set for training a machine learning model. And then, Beau, the really brilliant insight that you had was taking all of these features, which is already a rich proprietary data set. And the way that it's so well organized is mind-blowing to me. It must have a huge amount of work went into organizing all these inputs into a machine learning model. But then in order to be able to use all of those inputs, you could use an unsupervised machine learning approach that doesn't have labels, and you could kind of end up doing an exploratory analysis that would group related kinds of beers together based on these feature sets. But ideally, for brewing the most delicious beer possible, which is probably what a brewer is typically trying to do, it'd be great to have a label as to how delicious this beer is, and you obtained that. So you have this outcome, this label for all of these hundreds of rows that's based on consumer feedback. So it's a score rating, and there's a proprietary nature to this, but so I'm not going to go into too much detail of the scale, but it's a numeric score that is consistent across all of these beers. And so it allows you to have a rating of what people think of the beer, how delicious it is. And so for these several hundred rows, all representing beers, you've got tons of rich features going in, and you've got this outcome that you can predict with a supervised learning model. And so you blended these model inputs with this feature set, and you've been training a machine learning model. And one thing that I'd love for you to dig a bit more into some of the key features. Obviously, we can't go into all of the dozens, and to some extent, you might want to keep some of it proprietary. But giving us a sense of what some of the key features are in the inputs to this machine learning model. But then the second thing is that I'd love for you, after you've explained the features, to talk about the machine learning process. And something that's really amazing for me is that we haven't talked about this since you and I have been in the conversation. I would have mentioned it quickly in the episode's intro, but you don't come from a traditional kind of machine learning background. You weren't a statistician before. You weren't a computer scientist before. You I said you were a football player. You had an opportunity to go pro and play for the Rams in the NFL. You come from a family of football players, so you played college football with your two brothers. Your dad was a professional football player for the Washington, at the time, what they're called the Redskins football club. And so yes, you come from this football background. You studied psychology and sociology in college while you were playing football, and then you became a brewer because of your passion for brewing. But I guess in recent years, because of your love of science, you became interested in AI, and so you have been self-studying machine learning, and you have obtained some credentials, but not like getting a degree. So you have a certificate in the practice of data analytics from Ohio State University, which is cool. And I understand that you have more ambitions to study more. But the point that I'm getting to here, and I've been talking for a very long time, but the point that I'm getting to is, and I hope this is inspiring for any of our listeners out there, if you are interested in using machine learning in some application area. So in Beau's case, he happens to be passionate about beer. And so he was like, how can I use machine learning and AI to brew better beer? And he went out, he started listening to this podcast, to the Super Data Science podcast. He started studying things online, and having reviewed your code and gone through your process with you, I can say it's amazing. It is as good as the professional data science work that I see on a day-to-day basis. And well, I can't wait to find out what the results are like. But yeah, so I've now talked for a very, very long time. I don't know if there's anything else about your background that you want to talk about, but your background is something we'd love to hear about, the features that are going to this machine learning model, and then of course talk about the machine learning model itself.
Okay. Yeah. So when I first came up with this idea, I was like, man, this would be freaking awesome. I think this was when we started seeing Dolly come out, and I was like, I just need to start this project up. So I started gathering my all the proprietary data I had from brewing recipes. For the past, I think I was doing it for about 7 years then. It took me 6 months to clean all the data.
Wow.
So
I'm not surprised. That's something that's one of the things, I didn't know that, but it's one of the things that blew my mind about this whole project, was I was like, it's mind blowing to me that somebody had the foresight to be like putting all these data together over the years in this structured way, but that wasn't the case. It was retrospective mostly. So I imagine now you do a better job of like, okay, we brewed a new beer, let's get it into the right format, into this data set.
Exactly. Yeah. So that's actually why it's funny, because it's actually pretty similar to brewing in that, it's like 80-90% cleaning, and then 10% is actually executing, like actually brewing, which is definitely like true when it comes to this. That's where like these two things definitely align.
Well, so when you're making beer, this isn't something I was aware of. So our listeners who are data science practitioners will know that a huge amount of being a data scientist is cleaning the data, and actually training the models is typically a small part of the job. But so when you say cleaning in a brewery, like you literally mean like, okay, we finished brewing this beer, you just need to spend tons and tons of time cleaning everything and getting it back in spick and span before you start over?
Yeah, I mean, like the tanks and stuff,
Right.
the ground, the outside of the tanks,
Wow.
all those were. And now, granted, in a professional brewery, it's a little easier, but still, it takes a lot of cleaning and maintenance.
Whoa.
So, yeah, a majority of it is cleaning, making sure everything's sanitary, that sort of thing. So, yeah, when I came up with the idea, it had to work with. So when I was setting up the data column, the feature columns, there actually is 180 columns total.
Okay, right. I had this idea in my head that it was like 200, But when I just was skimming over it now,
it's about 200. But what I'll do is when we put guardrails up, we'll, depending on which model we're using, we'll shrink the data, the training data, or just keep it the same. If we're using Behemoth, which is the model with every single bit of our data, or if we're doing Chris P is my logger model. Chris P, we'll narrow down the rows so it just includes loggers, and then we'll shuffle around and delete out some of the feature columns so that it's all related to loggers only. Where in Behemoth is just like, okay, how do I get from A to B to the most customer satisfaction with no disregard to style or anything? So that's what Behemoth is. And usually what comes out the other end is like 10 to 15% alcohol beers, and a lot of stout influenced beers, but most of them are pretty off the wall crazy, and we'll try to navigate it to the ones that are practical to brew. So the one from Behemoth, for instance, that we'll be brewing here for the first time is I think it's a 8.5% Baltic Porter, which is a lager strain. It's got like 20 different malts in it. There's like dashes of certain malts, which I find fascinating. There's like lactose, marshmallows, vanilla, 3 different types of fruit. It's got a big whirlpool charge. Actually, just a normal like whirlpool charge of hops, which is really fascinating.
What does that mean, whirlpool charge? Everything else kind of made sense to me, but
the whirlpool charge is just like a dosing of hops in the whirlpool stage of the beer. So
yeah, and what's that?
So we'll hook it up in the kettle and we'll circle it after it's done boiling. And at that point, you can actually cool the wort down. So we'll cool it down to a certain temperature and then add hops if we need to. Usually whirlpool charges happen for IPAs only. So this is why it was fascinating to me that like a model like Behemoth throws a completely novel way of brewing a Baltic Porter, especially at me to brew. And this is where I think like this is like really novel stuff. Like this, some of these styles they're not anything I've ever seen before. And as a domain expert, I guess you could call me, just from brewing for several years, or 7-8 years now, I can look at a recipe that it generates and be like, wow, this is doable, and it sounds pretty nuts, but I want to brew it and see how it goes. So there's that generative. I don't know if I would call it a generative AI, but it's definitely supervised.
Yeah,
it's definitely supervised machine learning.
Yeah, it's typically we would reserve generative AI for something that's like generating like tokens, like natural language or code or something. It's typically what that means, but it is generative in the broader sense, in the sense of it's helping you generate new beer recipes. And I mean, and that's a key part of your machine learning process here, is that you have a simulation stage.
Yep.
Where, and I guess that, I guess maybe you could kind of consider that generative. So there's a process in your algorithm where you'll generate, say, 10,000 or a multiple of 10,000 random rows that are permutations of all of the relevant features. So, I guess for Behemoth, you have all 180 features going to the model, and so your simulation stage is creating 10,000 or more random permutations. But another really clever thing you did, and this leverages your domain expertise, is you set reasonable boundaries on those simulations. So in that sense, it's kind of like what a Bayesian statistician does. So a Bayesian statistician will have these priors around parameters or in their model that kind of constrain how far a parameter can go away from. So you could have like a distribution that describes the probability of where a parameter could be, and so that's similar to what you've done here with your simulation. And yeah, so I'm kind of jumping the gun here, but you have the simulation stage, and then later you're able to use the supervised model to say, okay, with these 10,000 simulated recipes based on the constraints that you have, what is the likely rating that a user would have based on these inputs?
Yeah.
So I don't know if I cut you off or if you want to get into a bit of what that supervised learning model is like, because you've tried a bunch of different approaches. I think it would be kind of cool for us to, if I haven't interrupted you too much and if I'm not changing the topic too much, it'd be cool to go into like the specific libraries that you used in order to create your supervised learning model.
Yeah, definitely. So like Darwin, as the name suggests, that's our fruited sour model. Darwin is completely, it's all genetic algorithm. So it only uses genetic algorithm. So Teapot is a really good way to utilize genetic algorithm. And what I'm assuming you might have come across genetic algorithms before.
Yeah, I mean, I can talk about it a bit if you like. So the idea with genetic algorithms is that you have you start with a random starting point with a bunch of different models, and then you see how they perform. So you have this like random starting point for a bunch of different models, and you see how they perform at the task that you'd like them to, and you take the top performers, similar to a way you might if you're trying to make a fast racehorse, or you're trying to make a yeast that ferments at a really low temperature. You'll take from all of the possible species. You can call these random starting point algorithm species, and you take the ones that are great performers. So just like the horses that are fastest, or the yeast that brews at the coldest temperatures. You'll take in this case, the models that are best at accurately predicting how people will rate a beer. And so that's like your first generation. You've now taken these top performers and you breed them together. That's the key thing with these genetic algorithms is you then say, okay, let's take two top performing models from my first generation, blend them together in some way. So there's some attributes from one parent and some other attributes of the model from some other parent, and you blend them together to get this second generation. And some of those second generation ones, you're going to end up having blended the wrong parts of the parents together by chance, because it all happens by random assortative mating, just like in the real world where chromosomes are sliced and diced at random to create a child. And so you're going to get in that second generation some that perform about as well as the parents did in the first generation. Some are just going to completely bomb because they got the wrong parts of the model process from their parents. But some of them are going to outperform because they happen to get the really good stuff that their parent one had and the really good stuff the parent two had, and they blend together. And so in that, the second generation is going to result in some child models that are better performers than anyone in the second generation. And then we just repeat this process over and over. So you take, just like breeding horses or making big apples or having cold fermenting yeast, you're picking the ones that are the best from each generation, mating them together. And yeah, so that's cool. So you call your genetic algorithm Darwin. And the key thing here, I think one of the key takeaways, other than in general how evolutionary algorithms work, is your recommendation to use the Teapot library, which I presume is a Python library.
Yeah, it's a Python library. It pretty much sets it up for you, just lays it up. So all you really have to do is define the Teapot algorithm, your training data, and then your parameters are really easy to find or to adjust. on the fly. So if you want to mess around with your offspring or a number of, I think it's mutation rate, you can definitely do that. And I found for teapot specifically, that genetic algorithm, it has a really solid like default set of parameters that were good out the gate. So if people are interested in genetic algorithms and they don't have a ton of experience like I do, it is definitely cool to learn from and utilize teapot.
Nice. Very cool. Yeah, I've actually never used an evolutionary algorithm on a project, so it's cool that you got to do that. And then you also, I think you made extensive use of the PyCaret library, if I remember correctly.
Yeah, I use PyCaret. I use H2O Auto ML as well. This is just for exploratory. So I use PyCaret and some other Auto ML just to like find some base models. So just to get an understanding of what works best on these on this training data. Most of them seem to be actually. I've been getting, a lot of them. I've been getting boosted trees, so I'll take a look at that. And for instance, if it's a boosted tree that comes out on the other end as the best score, I will take that and then run a separate model on that specifically to do a grid search and find the optimal parameters that way as well. So I'll brute force it.
Yeah, fantastic. So to break this down a little bit, so you use tools, AutoML tools like PyCaret and H2O, both of which I will put in the show notes, to allow you to identify models that might be useful for, say, whether it's Behemoth or CRISP. So Behemoth working across all of your features, or CRISP working with a subset that are relevant only to loggers, and also the data set sizes being quite different there. So Behemoth, there's a lot more rows to work with, whereas CRISP is only working with a logger data, which turned out to be a relatively small proportion. And something that I was kind of like, I was like, I don't really like IPAs. And you're like, well, dude, almost all of the beers that we have are IPAs. So we're going to have to reduce down the data set quite a bit. And so you end up in those different scenarios, different kinds of models are going to end up being great. But we don't know typically as the human, in this particular scenario, okay, if I've got the logger situation with a much smaller number of rows of data and a somewhat smaller number of columns of data, for this particular circumstance, does that mean I should be using the same model as I was for Behemoth? And maybe you could spend a lot of time messing around with different kinds of models to figure out what the best approach is for Behemoth in the first place. So to sidestep all of that, you quite cleverly used AutoML packages like PyCaret and H2O in order to be able to home in on what a great modeling approach would be. And as you're saying there, boosted trees ended up often being the approach, which isn't surprising. People who know from data science competitions like Kaggle, these kinds of boosted tree approaches like XGBoost end up often being top performers. And it looks like from your code that you're typically using scikit-learn as your trees as the library for finding for finding a trees module.
Yeah. Definitely use scikit-learn quite a bit. I don't know. Every now and then we'll come across some random one that scores really well, despite like using H2O and PyCaret, but they'll both score some off the wall algorithm that I haven't seen before or heard of before, like OMP. Which OMP, it's a greedy algorithm. I forget exactly what it stands for, but I thought it was a fascinating algorithm. But every now and then, yeah, you get some wild algorithms. What I do sometimes is, okay, so I'll do the AutoML run, I'd be like, okay, for instance, XGBoost did great on this, so we'll move it to the brute force method and use grid search. And then I'll experiment and ensemble that with something else. So one that I've really enjoyed using a lot is Tab PFN, which is a neural network, which is pre-trained on tabular data. If I'm correct, I might be saying
you are
that
correct.
wrong.
You are correct.
But,
Absolutely.
okay.
Yeah, I looked into this when you first mentioned it to me because I had not heard of Tab PFN before. But Tab PFN, so typically neural networks are not good at working with tabular data. And in fact, up until you mentioned this, I was not aware of people using neural networks, deep learning, for tackling tabular data problems. So deep learning, I mean, I wrote the book Deep Learning Illustrated, and I've taught deep learning tons of times. And I had students who regularly, as I was teaching deep learning courses, intro to deep learning courses, they would be interested in tabular applications. And I'd be like, yeah, maybe this isn't, maybe you can give it a shot, but it's probably not going to be very fruitful. You're probably better off using decision trees, for example, a boosted tree approach like XGBoost instead. So deep learning is usually better for pattern recognition, where there's a lot of structure in the data, patterns that can be learned kind of spatially in a way. So it's easiest to understand with machine vision problems. So when your inputs are images or video, there's a lot of spatial patterns that deep learning algorithms are adept at being able to tease out what the most important signal is from amongst all of that noise going into the model. And a similar kind of thing ends up happening with natural language processing, which deep learning has also proved, as we can see with the large language models that use deep learning under the hood. That are, it's not even under the hood. Fundamentally, large language models are deep learning networks. And so with natural language processing, with machine vision, we see amazing, powerful real world applications in machine vision and in generative AI, for example. But tabular data, where the rows are typically, they're not necessarily related to each other in the way that the pixels in an image are, or the words in natural language are. And so, up until you mentioned Tab PFN to me, I wasn't aware that somebody had come up with a way of doing neural networks effectively on tabular data. So it's really cool that you found that. I'm really glad you brought it up. And yeah, you probably have more to add.
Yeah, I love Tab PFN. It's limited, so it's limited on the number of feature columns you have, as well as rows. But so is my data. You know what I mean? My training data is not massive, so it's actually great at ensembling with other algorithms to either to kind of hone them in to be more accurate. I have found so that as an ensemble has been pretty eye-opening, and the fact that it's like instant. Tab PFN is like literally takes like half a second to train, and then and to make an inference. It's super fast, mind-blowingly fast.
Yeah, I guess with the relatively small amount of data points that you have, it's not like with the other approaches, like when you're talking about doing the boosted trees, and particularly using the grid search CV method out of the model selection module of scikit-learn. When you do a kind of grid search like that, you're going to be running so many different models. It's going to take a long time. The cool thing about deep learning, and part of what makes it so powerful, is that it allows for features to interact with each other in all kinds of crazy nonlinear ways. And so it's kind of like you're running lots of experiments on especially how nonlinearities and interactions relate to your outcome in a deep learning model. So it's cool to hear that it's so efficient on the relatively small amount of data in Tab PFN, and it doesn't surprise me that that's the case.
Yep.
Very cool. Yeah. So I hope that gives our listeners, obviously we, it's not like we can share Jupyter notebooks with people, unfortunately, or go into too much more detail on what you're doing, because otherwise you'd be giving away the secret sauce. But, some really great pointers here, general kind of machine learning ideas, Teapot for evolutionary models, PyCaret and H2O for AutoML, scikit-learn for a lot of your machine learning implementations, including, as you mentioned, boosted trees. I know also that sometimes k-nearest neighbors algorithms did come up as a top model in some of your situations. And TabPFN, super cool recommendation for fitting deep learning models to tabular data. So I'll be sure to include links to all of those Python software libraries in the show notes, and all of them are open source. So you can pick them up right today and start playing around with them.
So another one is called Lazy Predict. It's an amazing Python library, and this goes along the same toolage of PyCaret and H2O in order to find like a base high scoring base algorithm to work with, and it'll list them out and show you the top scores. And usually what I'll do is I'll run 2 or 3 of these AutoML algorithm search utilities or libraries, and I'll usually run those side by side to see, okay, this is actually legit. I should be doing XGBoost, or I should be doing ADA or linear regression, even though haven't seen linear regression pop up ever, but who knows? So anyways.
Very cool. And so in some of so you and I have had a few meetings, and so we were like, okay, let's brew. Like I agreed to help out and give you a bit of feedback on what you've been doing, the great impressive work you've been doing on your data collection and your modeling. And so we were like, okay, so we're going to have this collaboration beer. And as I already mentioned, I was like, I don't really like IPAs typically, so let's remove them from the data. And so you've been working on a logger. So we reduced the data down to just the loggers that were in the data set, and you trained. So you use these AutoML processes to identify crisp P, your logger only model, supervised learning model for predicting how people would probably taste one of your tens of thousands of simulated recipes. And so how's that coming along? Where are you at the time of recording? Where is the beer? And I guess we can tell people the name of the beer.
Yeah, I think so. So I tasted this beer yesterday for the first time, and out of all the lagers I've ever made, it's up there. It might be the best, like the best lager I've ever made.
Wow, that's unreal. Wow, I'm so happy to hear it. I was worried that we'd get on air, do all this rigmarole, and then you'd be like, we got to go back to the drawing board. We should have been using k-nearest neighbors instead of boosted trees after all. Why did I ensemble TabPFN? Because we can talk about this in a bit more detail. When you and I were looking over the results that came out of your proprietary supervised learning process, so you had these tens of thousands of rows simulated using the parameter constraints that you had on inputs, mm-hmm. and then you had your crisp P supervised learning model go over all of those 10,000 simulated rows and predict what people would rate. One of those came out by some margin at the very top, and that's this beer that you've been brewing. But there were some things about the features in it that, as I was, to me, it doesn't mean anything. When I'm looking at the inputs into your model, like most of them don't make any sense to me. I'm like, okay, I get like the alcohol content, like that one makes sense to me, and I can kind of through going over your sheet a few times, like I've learned, okay, like these are the hops, and like this is the mass of hops, and this is, but like that doesn't mean anything to me. Like I can't infer how that could possibly relate to the way that a beer tastes. But so as you were reviewing this lager that came out number one, by far in terms of how consumers are predicted to rate it, you saw some unusual things. Your eyes kind of lit up. What are those kinds of things that you saw?
Well, the main thing was Maris Otter. It called for, which is really fascinating because a majority of lagers do not use Maris Otter. That's mostly bound to stouts and pale ales and IPAs. So I thought that was really interesting. Not too crazy, but I thought that was fascinating. There was a really, again, a really large hop addition, and large for a lager, a large hop addition in the whirlpool. Not a ton of hops in the kettle, and no dry hopping. And in addition to that, there was also it called for sugar as well, a very certain type of sugar, which was called it's a D45 candy syrup. So we threw that in as well. And I forget what else. I'll have to take a look at that later. But I think there's different types of malt in there. Nothing too crazy, but I think there was wheat in there as well. So, anyways, as I'm brewing this and throwing candy syrup in it and doing the weird whirlpool charge, I was like, this is so bizarre, but in a really cool way. I feel like there's like an alien telling me what to do. And it makes sense. Like, it's just, I just know that like a person would never suggest brewing this, like usually. But it's coming out amazing. It's so unbelievably smooth, really light, and has a nice like depth of character and flavor. But and it's just, as I said, super clean, easy to drink. So we'll go ahead. It's about done fermentation, so it'll be conditioning for a couple weeks probably before we tap it, and package it. So
Amazing.
I'm super excited about it.
I've had an ear to ear smile as you've been describing this. It's been such a wild process. It's quite different. It's completely different from any kind of project that I've ever been associated with. And I played an extremely small part in this. This is really your project. You've done all the hard work on this, and it's fantastic to see what's come out. But to that particular thing that you're describing there, and I think that shows the real value in what you're doing at Species X, at least on the silicon side of things. With these silicon species, it's just like you're saying, like an alien directing you what to do. And so, you've spent years now, particularly quite a bit of time over the last year, though, that doesn't include all the time you spent getting the data together. But, you've invested years in aggregate into data collection, modeling, and also just training yourself on the side. And for that to come together and produce this result, which I guess we still haven't had the final taste, but it's come along now that you have a pretty good sense of it. For me, the two most exciting points in this whole process were going over this top-rated result that came out of your Crisp P model, the beer that it selected, seeing your eyes light up as you talked about things like the Maris Otter, which is a particular kind of barley malt. And as you say, like a flavor that people wouldn't think to typically put in a lager, combined with things like the specific syrup that you mentioned. Lots of hops added at the whirlpool stage, which I was like, oh man, like I don't like IPAs, and now you're at it. Now the algorithm suggested all these hops, it ruined everything after all. And you're like, no, it's okay, because at the whirlpool stage, lots of hops, they won't create too much bitterness in the flavor like they would if they were added earlier on in the process. And so I was like, okay, cool. Like, let's go for it. This sounds like a beer based on what you're describing that I would like. And so that was one really exciting stage in this. And then the second really exciting stage is right now to be able to hear that it's working out, because you don't know for sure with machine learning models. And most historically for me, in fact, maybe 100% of the time, with any other machine learning model I've ever been involved with in more than 15 years of working on commercial machine learning projects, all the other times I can know what the results are going to be like right away. I can see them on my screen. Does this work or does it not work? But with what you've done, it was going to be a couple of months from like, okay, this is the recipe it suggested. It's like a marionette to Beau, like an alien telling you what to do as you go through creating this beer, and you don't really know how it's going to turn out until the end. And so for it to be seeming like it's going to work out is super exciting. And so it sounds like February 8th in Columbus, Ohio, at the Species X Brewery. If we have listeners in Columbus, Ohio, actually, I do know that we have some Columbus, Ohio listeners, and I'll be reaching out to friends that I have in the city. And some friends may even come from far and wide of mine to come on February 8th and try out this Maris Otter lager that your Chris P algorithm has suggested. Can we tell people the expected name of the beer?
Oh yeah, it's called Krohn and Borg. If you want to go into depth on that.
Yeah, so it's this is actually a hat tip to my sister Stevie for coming up with this idea. So there is, of course, a very famous French beer called Kronenberg out of a city called Kronenberg. And so it's a very popular lager, particularly in Europe, but you see it on a lot of taps in the US as well. It's a pretty common, kind of like you'll see it alongside Stella's and Heineken's. So yeah, get your Kronenberg beer. And then so the idea here is it takes advantage of my last name, Krohn. It helps people, nudge them in the direction of pronouncing it properly, which is nice for me. And then it also adds in, if people are familiar with Star Trek, the Borg are these robots that become a big problem in the Star Trek series, specifically like the one that I grew up on, Star Trek: The Next Generation, in the 90s. They're kind of like the arch nemesis to Jean-Luc Picard and the Star Trek. So, and they're robots. So it's this idea of AI blended with humans, Krohn and Borg, and kind of rolls off the tongue. I wanted to call it Kronenborg 2064 to play on they called it Kronenberg's 1664 for their lager, but your marketing team said that might be cutting it too close.
Yeah, it might be a little too close. Yeah,
treading on trademark issues. But that would have been a funny thing. But yeah, Krohn and Borg. I'm so excited, Beau, for February 8th to come out to Columbus and try it out. And we will also I'll be recording a shorter episode, probably a Friday episode, while I'm there. Getting some feedback on the beer and answering maybe people's data science questions together to round out. So there will be an episode coming in the future on how the Kronenborg beer turned out from this amazing project that I am so grateful you invited me to play a small part in. Thanks a lot, Beau.
Yeah, I appreciate you to take the time to collaborate on this. It means a lot. And like you mentioned, I'm mostly self-taught in this. I have a little bit of formal education from OSU, but nothing super complex and over the top. So having you to come in and validate that what I was doing was, the correct way to do things, and making amazing suggestions into possible future iterations was really helpful. So I'll always be grateful for your time.
Yeah, my pleasure. And I'm definitely down for more of these. It's very exciting for me. It's, I'm passionate about beer. I love it very much. There are a few things I like more in life than beer. It's a funny thing. Like, people will be like, getting really fancy bottles of wine or whatever at dinner, and I'm like, just pour me like a little taste because you're wasting that expensive wine on me. Just get me a beer. Now that said, I can be a bit of a beer snob. But yeah, super, super honored to be part of this, Beau, and I'm glad that it seems to be working out so far. I was, up until today, I didn't know how this was actually going to turn out potentially in terms of flavor. We still don't know for sure, but I was just like, I didn't want to get my hopes up. I knew that there was a chance. This is an experiment, and so I guess I still know that there's a chance that it could fall down. But it seems like things are coming together, and so I've been kind of like holding my breath to find this out. So it's a really exciting day for me. I'm gonna be over the moon for a while for sure. One last final thing before we start wrapping up this episode, because you had an interesting insight for me before we started recording, and it shows how deep you get in the weeds on anything that you're thinking about. Is you had an interesting parallel between we talked about how when you were a college football player, you were also studying psychology and sociology at Virginia Tech. And you made an interesting parallel before we started recording to the large language models like GPT-4 that we have today.
Yeah. So this actually relates back to the project that we're doing here too. I've been using an agent, open source code that will go out and do research utilizing Claude or GPT-4 or one of the big ones.
That's an open source agent?
Yeah, it's Baby AGI.
Baby AGI, yeah.
So that's a good example. It's a iterate. It's like riffed off of it. So it's not exactly Baby AGI. It's my own repository that we use here in house at Species-X.
Oh, cool.
But I can't like open source it just because there's a lot of proprietary information inside of that repository. So yeah, it's definitely a riff off of that. There's a lot of, in addition to changes on the actual Baby AGI code, there's also a lot of prompt engineering. And I still can't believe I'm saying the word prompt engineering, but it's definitely a thing now. It's just crazy to me. So so what the goal of the agent is to go out. It's given a goal. It goes out, does research on the top beers, and then it'll create a brand new recipe, and then it will go ingredient by ingredient, improving the recipe at each stage. So it'll keep going until you just tell it to stop. But yeah, and then in addition to that, it'll tell me its name. And then it also prompts me with an image of itself to put into a image generator. So going off that's you'll see on the website, it says various agents. That's what we're talking about there. We'll eventually, I have to figure out how I'm going to release one of those agent beers, but in there you can see, even in the Baby AGI repository code, it'll say, temperature, and you can mess with the temperature in there. I've noticed when you up the temperature of the model, it tends to do some pretty crazy stuff.
Yeah, and so to dig into this is when you have a large language model, there's a key hyperparameter, so a key attribute that you can adjust called temperature. And I guess it actually, in a way, we can bring this analogy back to your sweaty yeast thing, because it's kind of like, when you have cold water that you're brewing in, the reactions are slower, more predictable. And when you're doing the reactions like you might with an ale, in a warmer temperature, the action proceeds more quickly, but it's more sporadic. So it's similar with temperature as well. So actually, if you turn the temperature on a large language model all the way down to 0, you will end up with a totally unstochastic model. It'll be a deterministic model where every time you run it, you'll get the exact same output. So it's just predicting the most likely generative sequence, whether that's an image or text or code or whatever you have your generative AI model doing, whatever you have your large language model doing. It is you turn the temperature up, it will have more random outputs. So it will explore, so you'd end up with more creative images, or more creative, more random stories. And yeah, so that relates, Beau, to.
Yeah. So when I was studying psychology, you talk about how as a person gets more, when their schizophrenia really starts to develop, you can actually see this in artists too, that are developing schizophrenia. At first their drawings will be very, they'll look like very realistic or like photorealistic in some cases. They look normal essentially. But as the schizophrenia develops, you'll see like a lot more colors and more like subjective shapes and stuff, more abstract, till eventually it's completely unrecognizable and it's just all colors on a piece of paper and no shapes, no nothing. So like one example I saw was a person was drawing a cat and they were really good at drawing cats, and then eventually like the cat forms start to go away until it's just like blobs of color. And going back to the LLM reference, I noticed when I upped the temperature on it, it would like talk to itself. So it'd be like, it would have conversations with itself, and none of it made any sense. Like a word salad is what people call it. So it would just like, chirp off and start making absolutely no sense whatsoever. I was like, gosh, this reminds me of like schizophrenia. Like, it's just like strikingly similar to like the development of schizophrenia. If you have a temperature on 0, like you were saying, very proper, you know what I mean? Very, will not, do anything too crazy. But as you up the temperature, that cat, referencing back to the cat drawings just got crazier and crazier and crazier till it's unrecognizable and makes no sense.
Yeah, so it's an interesting analogy. And yeah, I mean, at a surface level, we're certainly not psychiatrists or experts in schizophrenia in any way. But from the abnormal psychology courses that I took as an undergrad studying neuroscience, I can see the parallels that you're making, and it's an interesting, and maybe it's an interesting parallel. And I wonder if somehow there is something useful there for schizophrenia researchers. I don't know. There may or may not be. Might not. I don't, like the way that we have animal models of diseases. So we'll use mice in particular, or sometimes rats, as animal models of different diseases, including psychiatric diseases. And so I wonder if someday we'll have machines that can substitute, and maybe even be a better substitute, because obviously mice don't have language, for example. But yeah, I wonder if there's people out there working on these kinds of application areas where you can have an LLM that models schizophrenia, and somehow that provides some insight into the actual biological disease itself. So yeah, interesting ideas there, Beau. Thank you so much for this fascinating episode. I hope our audience has enjoyed learning about beer and about how anybody can be training themselves to take advantage of machine learning and AI open source tools that are out there, learning opportunities that are out there that are available for free online. And yeah, so you can leverage your domain expertise to create some really cool AI projects like you're doing at SpeciesX. And yeah, hopefully we're going to find in a couple of weeks that Kronenborg is a delicious beer and that this project was a success. I'm super excited about it. Been a great day for me already hearing this positive news. Beau, as a regular listener to the program, you will be aware that I can't let you go without a book recommendation.
Yes. So I love space too, in addition to this. one of my favorite books I've read recently was Operation Hail Mary. I won't go into, I don't want to give too much away, but it involves space, microbiology, and saving the world. And
Okay.
it's amazing. It's a fiction book, but yeah, it's one of my favorite books I've read in a while. I think they're making a movie with Ryan Gosling starring it. So
Really?
That's something else. That's
that'll be big.
something to keep your eyes out for.
Awesome. All right. And Beau, so how should people follow you? Obviously, I'll include a link to speciesxbeer.com in the show notes. Where else should people connect with you?
I'm really active on Instagram. So at speciesxbeer. That's the same handle for all of our social media. So active on Instagram, very active on Threads. I'm here and there on Twitter and then Facebook as well, but mostly Threads and Instagram. You'll be able to find us. So.
Well, you get a prize for being the first guest to ever say that they actually use Threads.
Really?
Yeah. I've had people, especially when Threads first came out, they were like, I've created a Threads account. But yeah, you're the first one to actually say that they're using it. So that's cool. It's nice to know.
Now, I don't have a ton of followers on there, but yeah, I post there pretty frequentally, and I don't know. I have more followers on there than Twitter/X. So
nice.
I'm just trying to.
Nice. All right. Well, all of our many Threads listeners to the podcast, they will be checking that out right away. Beau, thank you so much for taking all the time today, and I'm looking forward to catching up with you in Columbus on February 8th.
Thanks for having me, John. I appreciate it.
Well, I'm super, super excited to try the Krohn and Borg Maris Otter Lager in a few days. Wow. Unreal to hear that Beau's AI beer project is likely to work out on the very first attempt. In today's episode, Beau filled us in on how sweaty ales have more flavor and efficiency than cleaner fermenting lagers, how he painstakingly curated nearly 200 feature columns for 300 beers over 6 months, how he used the PyCaret, H2O, and LazyPredict AutoML libraries, as well as the Teapot genetic programming library in Python for homing in on great models for his beer rating predictors, how he used scikit-learn extensively throughout his work, particularly the grid search CV method for finding optimal model hyperparameters, how he ensembled more traditional ML models like boosted trees with tabPFN deep learning models to get even better results, and how you can join us in person at SpeciesX Brewing in Columbus, Ohio on Friday to taste the resulting Krohn and Borg beer that emerged from these years of AI R&D effort from Beau. As always, you can get all the show notes, including the transcript for this episode, the video recording, any materials mentioned on the show, the URLs for Beau's social media profiles, as well as my own, at superdatascience.com/755. Thanks to my colleagues at Nebula for supporting me while I create content like this Super Data Science episode for you. And thanks, of course, to Ivana, Mario, Natalie, Serge, Sylvia, Zara, and Kirill on the Super Data Science team for producing another delicious episode for us today. For enabling that super team to create this free podcast for you, we are, of course, so deeply grateful to our sponsors. You can support this show by checking out our sponsors' links, which you can find in the show notes. And if you yourself are interested in sponsoring an episode, you can get the details on how by making your way to johnkrohn.com/podcast. Otherwise, please do share, please review, please subscribe, and all those good things that help us get the word out there about the show. But most importantly, just keep on tuning in. I'm so grateful to have you listening, and I hope I can continue to make episodes you love for years and years to come. Until next time, keep on rocking it out there, and I'm looking forward to enjoying another round of the Super Data Science Podcast with you very soon.